Have you ever just stopped and thought about how a single cell, one tiny cell, can build an entire body, trillions of specialized parts, all from one starting point? It is, without a doubt, one of the most magnificent stories in all of biology. And today, we're going to dive into the fundamental principles that make it all happen.
Welcome to Silverlight Education.
In this lecture, we will be discussing the BZYCT 133 course, Block 3, Unit 10.
So let's get right to it. The big question, the one that has fascinated scientists for literally centuries, is right here. How does one cell give rise to trillions of others that know how to organize themselves into eyes, a heart, a brain? It's a process that is just absolutely mind-boggling when you think about it.
You know, what's so amazing is that this whole process isn't random at all, not even a little bit. Whether you're talking about a human, a fish, or even an insect, development follows this remarkably predictable set of stages. It's like nature has a universal blueprint it follows every single time.
So this incredible journey kicks off with fertilization, which creates that very first cell. Then comes cleavage, which is just this explosion of rapid cell division. After that, we have gastrulation, and this is where cells start organizing into three foundational layers. You've got the ectoderm for things like skin and nerves, and the mesoderm for muscle and bone. And finally, in organogenesis, these layers assemble into all our organs. It's precise, it's predictable, and it's just astonishing.
But you know, our understanding wasn't always this clear. Far from it. For centuries, a really fierce debate raged among scientists about what development actually is. Were new parts being created from scratch, or was it just a matter of making something that was already there bigger?
The whole debate really boiled down to two opposing camps. On one side, you had Aristotle's theory of epigenesis, the idea that new structures are formed progressively, kind of like a sculptor carefully shaping a block of clay. And on the other side, you had preformation, the belief that a tiny, fully formed being was already there from the start and just needed to grow.
And this preformation idea led to one of the most famous and kinda weird ideas in the history of biology, the homunculus. These scientists, the spermists, genuinely believed that a perfectly formed, miniature person was just curled up inside every sperm. It's a fascinating theory, for sure, but, well, better microscopes eventually proved it wrong.
Okay, so if we're not just inflating a tiny preformed human, how do all those brand new cells figure out their jobs? Let's zoom way down to the cellular level and find out how a single cell's destiny is actually decided. Think of a cell's journey to its final career as a three-step process. First up is specification. At this point, its fate is kind of written in pencil. It has a direction, but if you change its environment, you can erase that path and write a new one. Next is determination. Now things are serious. The fate is written in permanent ink. There's no going back. You can move that cell anywhere and it will stick to its destiny. And finally, there's differentiation. The cell actually becomes its specialized self, contracting like a muscle or firing like a nerve. But this whole mystery just got deeper thanks to two classic experiments with completely conflicting results. First, Wilhelm Roux destroyed one cell in a two-cell frog embryo, and he only got half an embryo. This suggested that a cell's fate was pre-programmed. He called it mosaic development. But then, Hans Driesch completely separated the cells of a sea urchin embryo, and each one grew into a complete larva. This pointed to the exact opposite, that cells could adjust. So which was it? Fixed or flexible?
And that contradiction leads us straight into the ultimate puzzle of modern biology, a puzzle that's hidden in our very own genetic code. The whole puzzle really starts with this one core concept, genomic equivalence. Now, what that means is that basically, every single cell in your body, a skin cell, a liver cell, a brain cell, contains the exact same complete set of genes. The entire genetic library is in every room of the house.
So, let that sink in for a second. If a skin cell and a brain cell have the very same DNA blueprint,
Then how on earth do they end up being so radically different? I mean, in their shape, their job, everything. This was the central mystery we had to solve.
The ultimate undeniable proof of genomic equivalence, and a huge clue to the answer, came from cloning. I'm talking, of course, about Dolly the sheep. When scientists created Dolly from a single specialized adult cell, they proved that even that one cell still had all the genetic information needed to build a whole new organism. No information gets lost.
So if the information is all there, but the cells are different, something else has to be going on, right? It's time to reveal the solution to this puzzle, the master switch that controls what every cell becomes.
The easiest way to wrap your head around this is with a really simple but powerful analogy. Think of the genome inside every one of your cells as a giant library. It contains every book ever written on how to build and run your entire body.
Now, a skin cell walks into this massive library, but it doesn't browse around. It goes straight to a specific aisle and only reads the books titled How to Be a Skin Cell. It completely ignores all the other sections.
And you guessed it, a brain cell walks into the very same library, but it heads straight to the neurology section. It only checks out the books on how to be a neuron. Same library, different reading lists.
This library analogy perfectly explains the real scientific principle. It's called differential gene expression. This is the master switch. It's the process where cells with the exact same genes use or express different sets of those genes. They turn specific genes on and off to create their totally unique identities.
So let's quickly recap our whole investigation. We've seen that development is this universal blueprint-like process. We learned that a cell commits to its fate through these progressive, irreversible steps. And the central truth is that while all our cells share the same DNA, the real secret to all that diversity is which of those genes they decide to turn on or off.
It really is an incredible thought. Understanding how one cell builds a body reminds us that every single cell inside you holds the potential for an entire life. And that just leaves us with this tantalizing question. What other profound secrets, locked away inside our cells, are we just now beginning to learn how to unlock?
We will meet you in the next lecture.